Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other for solving the problem in the comment below. So here today we will discuss about the Eliza test. So what that means, Eliza test. Eliza test is made up of direct, indirect and sandwich Eliza test. So the Eliza mean enzyme linked immunosorbent assay. Enzyme linked immunosorbent assay we are using. It means the enzyme linked antibodies used for this detection of the antigen are present. What is the antigen? What is the type of antigen are present in the patient, and we can detect that antigen by the different methods and different mechanism. So to understand for this, here is the person which contain a infected antigen. So that antigen can be pathogen or protein or something which that is infected to give a disease. So that means infected person when take the blood and take a serum. Serum contain a antigen or antigen. Yes. So let's begin to detect the antigen first of all. So this serum contain a many type of antigen. For example, it can be A, B, C, but we are detecting the B antigen for understanding, for looking if either are present or not. So that means if the antigen B is present, so that is very difficult to identify with our own self, with our own eyes, because it is macromolecule, but it is not naked eye can detect it. So let's begin to understand. Antigen A, B, C, which that is the type of pathogen. So we will detect about that. But there are many antigen which that is used, but we will take the specific antibody which that is prepared in the laboratory to detect with the enzyme linked antibody. So here is the 96 microvials are used for the detection of the antigen which that is present in our body. For example, if we are infected that pathogen, so we are hypothesis or we are assume that it can be infected the P antigen, for example influenza virus or HIV virus, but we don't know about that. So how to detect that antigen we will discuss in this method. So here we will take serum and separate the antigen from our blood, which that is it's during the infection and not produce the immunoglobulin against the antigen still here. So the antigen will be separate. So that means the ELISA plate is here. This ELISA plate is looking like this. So basically simple ELISA test, it is direct, we will discuss first. So the simple ELISA test, it means the enzyme linked immunosorbent assay used directly. So reagent in lab preformed, we can prepare the reagent and that antibodies immunoglobulin which that is used. So it means the different type of antibodies will be we can produce in the laboratory by the linking of the enzyme. So that means that there are a lot of antibodies are present. So here is the basically ELISA plate. So let's begin to understand the antibody A, antibody B, antibody C, which that is attached with the enzyme linked antibody. It is prepared in the laboratory. So it means here we want to detect the specific pathogen if present, so we can detect. If not present, so the enzyme linked antibody will not attach to the epitope of the antigen. So here we can looking for B antigen, for example, and we can prepare the against the antigen to produce the antibody in the laboratory by the linking by the enzyme. So the enzyme linked antibody, this antibody convert colorless substrate into colorful product. So it means we can detect by the by the spectrophotometer. So the spectrophotometer is show the spectrum, photo mean photon, light, and meter mean the measurement tool, measuring tool. So that means here we will discuss about the method to detect that antigen which that is present the epitope, and we will take the sample from our blood, from our the patient blood, and we can sticking in the polystyrene plate. It is known as the Eliza plate, and blocking agent is used for the blocking of the antigen, and it is sticking, and polystyrene plate, the plastic plate, is used to attach that antigen in the Eliza plate. So that means antigen antibody complex will be formed where the epitope is match the enzyme linked antibody which that is prepared in our lab to bind, but any other antibodies will not attach because epitope will not match. So that means the antibody enzyme linked with the antigen B will be bind, and it means it is enzyme linked antibody bind with the antigen, but other will not other mismatch, and it will remain unbound. So that means it will remain unbound when apply the antibody B which that is prepared in the laboratory. It means that we are linking the enzyme with the antibody B against the antigen B. For example, influenza virus or coronavirus or something, you can think about that. After that process, the next step is the washing. This washing step, which that is wash away the all antibodies which that is not useful for the epitope for B. So that means so every antibody will be washed away which that is will be not bound, but those bound will remain behind. So that means here is the substrate which that is used and to produce the colorful product. We can see also check concentration of color. We can also buy spectrophotometer for coloration. So the quantity of coloration and the quality of coloration will be variable. For example, this simple Eliza test is the direct because it is directly we can detect. So that is unbound will be wash away and antibody basically wash away. So it contain advantages and advantages. It means two basically beneficial we will discuss also. So the first of all we will understand about the advantages and disadvantages. So first of all, the quick way to perform test. So it is quick way to perform test because it is very simple, and simple Eliza test is a direct and we are not using any other things. And disadvantages is that sometime antibody may not react with antigen, so the number of enzyme can be less for color. So that is the disadvantages and advantages. So let's begin to understand the indirect Eliza test. Why we call indirect Eliza test? Because we use another animal to proliferate that antibody, and we use the another animal. So for example, here is I am drawing the indirect Eliza test. We should understand about the secondary antibody uses with enzyme, but primary antibody is not used with enzyme. So let's begin to understand. We are detecting the antigen, pathogen, and here is the A antigen, B antigen, and C antigen, which that is the different type of antigen which that's come from the lab, that is the epitope. But we take the antibodies from the patient which that is produced against the pathogen in our body. So that means the serum contain immunoglobulin and no enzyme linked because it is come from the patient. So the primary antibody will be bind with the B antigen, so we can recognize that it was the pathogen is B. So we can detect that it is the pathogen B. So that pathogen B we can classify, we can adjust and understand about that, and we can say that was the disease which that is produced due to the antigen B. For example, the pathogen B. For example, the coronavirus or something, you can say. So the primary antibody is not enzyme linked, but we can enhance the coloration to check more specificity and very qualitative test. So the now we are using the amplification of coloration. So we can use the another animal, and that antibodies which that is bind with the B antigen, we can inject in the animal. So when inject into the animal, so the foreign antibody inject into the animal, so that means it is a foreign antigen antibody, so it is not a good thing for that animal. So for example, the human antibody IgG, which that is important for understanding, so that is the exemplification. So the immunogenic parts is basically activation. So that means antigen role foreign contain a epitope. So there are many epitope are present into the foreign antibody. So animal produce the antibody against the antibody of the foreign which that's come from the patient, and that's against the antibodies is epitope. Against the epitope, antibody will be produced by the B memory cell of the animal. So let's begin to understand. So immune system of the animal basically is activate and bind the epitope of the antibody of the foreign patient to bind with the animal antibody against the epitope. So that is known as the polyclonal antibody, while the monoclonal antibody is also produced by the hybridoma technology system. So that is the separate things we can understand in another video, but I have also made that video, so you can search that, or I will give the link in description below. So this was the indirect Eliza test. So let's begin to understand. That is the epitope is produced, and that is antibody against the epitope will be produced. This antibody can link with the enzyme in the laboratory. So important thing is that here is we take the antibody and inject into the animal to produce the epitope containing epitope antibody complex, and so that is basically we will inject into the animal and sacrifice the animal or something, you can say, and separate the from the blood of the animal, and that means the enzyme will be linked by the process of the laboratory. So that means enzyme linked epitope antigen antibody complex with the primary antibody to the secondary antibody. So the enzyme linked antibody is known as the secondary antibody enzyme linked. So that means amplification of color system by the using of substrate will be more as compared to the single primary antibody. So that means we are clear the disadvantage of the simple Eliza test into that indirect Eliza test for amplification of the coloration. So the amplification of color system after modification, more color production are more sensitive and more specificity is occur due to the qualitative. That is the test is qualitative, and we can watch by the spectrophotometer. So the spectrophotometer is used for the measurement of the color. So that is substrate used for it. For reaction with the enzyme. So the enzyme binds with the substrate to give a product. That product will be colorful. So it means the color will be show. So next is the third one is the direct and indirect sandwich ELISA test. So the direct and indirect sandwich ELISA test, what that means? Sandwich is what that means. The sandwich means you know about the bread, like structure which that bread paste the cheese and more bread will be paste, that is known as the sandwich. So just like that. So looking for special molecules, we can check the special molecules into the patient. For example, if we are looking B antigen or molecule or some hormone or some changing of the hormone, to check that or check the either testosterone are present in our body or not, or some antigen are present in our body or not, we can check that. So if we will take the sample from our blood, for example a patient, so that's antigen containing blood. So that's antigen, basically B antigen, we suppose B antigen. So the microvial is basically pre-coated antibody B antibodies, which that is pre-coated, and we can detect that. So against the antigen, antibodies already done in the laboratory is the pre-coated antibody B. So that means antigen is here, which that is antigen apply into the pre-coated antibody B antibodies, and that means antibodies stick into the polystyrene plate of the plastic. So that means the reagent used is the reagent lab pre-formed basically sticking and blocking agent is used also. So that blocking agent will not allow to remove that antibody, and that means antibody as present is here and antigen are present. Antigen mean we are prepared against the antigen B, which that is our interest to check. So our interest is the antigen B, but we will take the sample and apply into this environment, and antigen B will be bind with the antibody, but more antigen are present in the sample, so that antigen will be not bind. So that means it means the more another antibody will be removed after the washing, and that means the antigen will also remove. So remember, that is important things for you to understand as well. But here is my mistake about that. The antibody is simple and BBB, not a C or another. So antibody is same. So that is why antigen will be bind with the antibody, and other antigen or other pathogen or other molecule will be washed away after the washing. So that's after the washing we will apply the another, that's laboratory basically we apply the enzyme linked antibody, which that is against the antigen another epitope, and that is bind with the enzyme linked antibody with the antigen, and that means that substrate will be used, and after this substrate will be using, first of all we will washing away. That means the another enzyme linked antibody, which that is not specific for the antigen B. So that means that will be not bind with the antigen B. So that means that antibody is not useful for us. So that means after the second washing process, we can separate the substrate. We can separate the other antibody enzyme containing antibody. So that means basically here is the coloration showing basically after the using applying the substrate to coloration showing by electrophotometer by after the washing away all antibody and enzyme linked antibody, which that is specific of the antigen B is bind. So that is sandwich like structure you can say. So this is known as the direct sandwich ELISA test, while the indirect sandwich ELISA test is also used for. So the coloration showing by electrophotometer is also. So the direct sandwich ELISA test is here, while the indirect sandwich ELISA test is another very interesting process. So here is the antigen antibody complex, which that primary and secondary antibody, two epitope containing antibody, which that's upper and down, and basically enzyme link antibody, which that is not here using in the upper sandwich like structure, but these antibodies contain epitope, but we can again that inject into the animal to produce against the anti-epitope antibody, and that antibody will be bind with the enzyme, so enzyme bound antibody complex will be formed. So this is done in the laboratory. So for example, here is we can say that here is the antibody enzyme linked complex, which that is used against the antigen, and that's epitope specific antibody, which that's applied to the indirect sandwich Eliza test. But indirect sandwich Eliza test is used the animal. So that means again that is indirect Eliza test which that is used. So the here is the indirect sandwich Eliza test. So the indirect sandwich Eliza test, the enzyme bound antibody will be produced into the animal, and after the separation into the lab we can attach the enzyme and bound with the enzyme, and it is secondary antibody which that is attached with the primary epitope of the antibody. So that means attachment of this enzyme apply the substrate in with a specific enzyme to give the coloration. So that color is detect that is the molecule is adjust and specific. So that is more quantity of color and specific and sensitive. So thanks for watching. Please subscribe the channel and watch please more.